colleagues in Glasgow, particularly Simon Fisher, who should have been giving this talk actually, but he's not available to attend the meeting this week, unfortunately. So today I'm talking about identifying protein complexes binding to risk functional variants in the uromodulin promoter. So previously we had identified RS1333226, I'll call it 226 from now on for speed, to be significantly associated with blood pressure. Turns out this was within 2,000 base pairs of the uromodulin gene in the promoter. Function of uromodulin gene has been ascribed to a few functions, including ion transport, immune function, and calcium signaling. The key thing I'd like us to take away from this first slide is that data I've not had time to show you, but we have promoter luciferase functional assays showing that the risk SNP and the protective 226 SNPs do not have functional effects on the promoter, whereas a close SNP within 500-600 base pairs or so in almost complete linkage disequilibrium, SNP RS499781, I'll call it 499 from now on, does have a functional effect. A couple of interesting features about that particular location as well. We have validated the association with blood pressure using the mouse knockout. Uromodulin is also known as tan-horseful protein, so this is the THP minus mouse. So on normal salt, the uromod knockout has a lower blood pressure than wild type 129 mice. It's also, if we add salt to drinking water, it's almost completely insensitive to this increase in blood pressure we normally see in wild type glutamates. So the purpose, as I said today, is to identify binding partners which associate at this functional SNP site and how they may affect differential expression using electrophoretic mobility shift assays, or IMSRs. I've got one slide that I hope to show you just on RNA-seq as well, actually, from human samples. I hopefully have time to show you that. Just briefly about the design, the principle is quite straightforward for this gel retardation type of experiment, where a protein bound to DNA will retard the DNA in a polyacrylamide gel system. So this oligo, in this case, is labeled with biotin, and it contains this SNP of interest. So the first thing we do is do binding of this labeled oligo with nuclear lysates, in this case from HEC293s. So we run the gel and we see that we have the unbound labeled oligo, and then what we're looking for is a protein that's bound to the oligo, which shifts the band up. The purpose then is that we would like to identify what these proteins are binding. Of course, if you know what you're looking for, you could add an antibody to the system, so you would have yet another shift in the band, which would be a super shift assay. But if we don't know what we're looking for, then we have to do mass spec. So when we did this, we had significant differential binding affinity detected at 499 SNP, not the discovery 226 SNP, and this was significant. So we prepared some IMSR incubations from the 499 and controls, purified the samples, and submitted for tandem mass spec. A total of 110 proteins were identified by mass spec. To ensure we had good identification, we ensured that the matches had at least 10 peptides, and in this case with mascot scores in excess of 200. So out of the 110, 47 of the proteins were unique to the preparation with 499, including the PRP1 protein. And with pathway analysis, we identified a protein complex between PRP1 and HNF1 beta, which has already been shown to be upstream of the uromodulin gene. And there was a protein complex which has been identified in the public domain, and therefore our hypothesis at the moment is that differential binding activity of the PRP1 at this locus may underpin the GBARS association. I think I have time to just quickly show this. This is an RNA-seq that we did in human renal samples, biopsy samples, where we're comparing humans with high and low UMOD expression. I don't have time to go into too much, but what I will say is that from the RNA-seq, we identified co-expression between Uromodulin and SLC12A1, or in KCC2 as it's better known. And we then went on to do qRT-PCR in just under 100 of our samples, showing an excellent correlation between these two genes. Simon also went on to show in some publicly available kidney single cell data sets that there was significant significant co-expression between Uromodulin and SLC12A1. So I probably don't have time to sum up. So this is what I've told you. But together, this data strongly suggests that the UMOD associates with blood pressure regulation through sodium natriuresis. And this is facilitated by co-expression mechanisms with the sodium potassium co-transporter. And this association between UMOD and SLC12A1 is the subject of one of our clinical trials that should be reporting by the end of this year. With that, it's a pretty picture of Glasgow. It was nice when you guys visited, and thank you to funding from the British Heart Foundation.
Me to talk about a new rat model we've generated that expresses Cre recombinase at the µ-opioid 1 locus. So the Yavin Shaham.
Tom, sorry, are you planning to share some slides? We can't see your slides.
Good point. I'll try again. There. Okay, so we generated a rat Cre recombinase model for Yavin Shaham at NIDA. They're interested in studying opioid addiction. So we dropped Cre recombinase into the OPRM1 locus in the rat. I think everyone in the audience is familiar with what addiction is. The µ-opioid 1 receptor mediates the positive response to morphine administration. And the general idea is if we can understand more about the function of this receptor, we may come up with new ways to help treat people who are addicted. So the addiction is a serious societal problem. And you can see from this review that people are dying from opioid addiction. And that's one of the reasons why NIDA is so interested in opioid addiction. This is the rat OPRM1 gene of the genes I've worked with. This one wins the prize for having the most transcripts. And I believe, yes, most of these transcripts do produce proteins. So we were directed by Huda Kiel's lab to work on the MOR1 seq transcript. So we took the last exon and we dropped in a 2A peptide and a Cre recombinase coding sequence just before the termination codon with Cas9 technology. So the general idea is you make a chromosome break in a fertilized rat egg, and then you give the rat egg an opportunity to repair the chromosome break with a long single-stranded DNA donor. When this happens, then the messenger RNA is going to be bicistronic. It will produce two proteins. It'll make the µ-opioid 1 receptor protein, and it will also make the Cre recombinase protein in the same neuron. And because this is a targeted knock-in instead of a plasmid transgene, the fidelity of expression is excellent. And Yavin's lab has developed preliminary data looking at messenger RNA. This is all unpublished. And what you can see here is in this brain section, Cre recombinase RNA labeled in red, µ-opioid receptor 1 RNA labeled in green, and the merge, you can see excellent co-localization of the two signals. We heard earlier from Dr. Meyer about palatable food self-administration paradigms. So Yavin has just finished testing the new rats to see how they perform for food self-administration, if they can learn the paradigm and if they behave normally compared to wild type. So they have a normal learning behavior. And then in terms of teaching the rats to self-administer food after different time intervals of signals, the rats have figured that out too. And now they are in the middle of doing morphine self-administration trials. So then the idea is you'll have a rat that can become addicted to morphine, can dose himself, and ultimately then one approach would be to use optogenetics. Now you have an addicted rat, you break the addiction, you take the rat off morphine, let him get unhooked, and then you bring him back into an environment where he can choose to self-administer morphine, self-administer palatable food, or self-administer social interaction. And can we watch which neural circuits in the brain are being triggered? What are the µ-opioid 1 receptor neurons doing? What other neurons are they talking to, et cetera? And hopefully this will give us some insight in how to better help people get off morphine addiction. And with that, I will take it to the acknowledgements. So I want to acknowledge the people in my group that made the genetically engineered rats. Yavin at NIDA, this is his baby. This is the rat he wanted to study. Huda Akil's group helped us in the identification of the gene to target. And then Bridget Kiefer gave us input. She's got a fabulous wealth of experience in mouse models targeting opioid receptors. So I'll stop there and take questions.
Lovely, thanks very much, Tom. Yeah, we do have time for a few quick questions. Abe's got his hand up.
How do you think about the breeding scheme that should be used? So Sprague dollies are outbred. And I understand that that's what Yavin probably wanted, was a Sprague dolly. But how do you think about the appropriate breeding scheme to maintain a mutant allele like that on an outbred background?
Well, if you're going to keep it on an outbred, I mean, Sprague dolly rats are, quote, outbred, you know, but they're all albino, right? So how outbred are they?
Yeah, they've got millions of SNPs. We've done a lot of work with them.
Yeah. So I think
quite diverse.
Right. So I would refer, you know, after three intercrosses amongst themselves, I would outcross them back to the Sprague dolly stocks that are being maintained at Charles River. We assume that Charles River is doing their best to maintain Sprague dollies in an outbred condition.
Okay, good, thanks.
Yeah, you know, standard genetics, right?
Thank you very much, Tom. That's great. Can we move on, please, then, to our next speaker? Suzanne Mitchell, actually, from Oregon Health and Science University. Suzanne.
Okay. Hopefully you can see that. Great. Okay, so I think this talk actually follows very nicely from Vivek's in many ways, because we're going to be talking about behavior. And this is a picture of me in my lab. You can see we're a fairly small lab, but these are the guys other than me in the red who do all the work. What I'm going to talk about is a behavioral paradigm that we use in decision making, delayed reward discounting. That's what people call it in humans. And it's questions where people choose between larger later rewards and smaller sooner rewards. So for example, $25 in 7 days versus $20 available right now. And usually there's a range of delays offered, so you can get a sense of the gradient of choices. And it's usually coupled with a reward that's available immediately. And I actually think that that's really important, but don't have time to go into that. So the reason why we're interested in this is that over the last 20 years, delayed discounting has been associated with all sorts of psychopathology, in particularly steep delayed discounting, meaning more choice of the immediate reward at discounting because of the delay of the value of that later reward. So we see a nice paper there about addiction. Here's something about obesity, and even more recently, a meta-analysis of psychiatric disorders. So one of the reasons that this group, I think, should be interested in delayed discounting is that data has indicated that it's heritable, both in human-focused data and animal-focused data. And in fact, the group's own Abe Palmer has done a very nice GWAS study led by Sandra Sanchez-Roger, indicating some particular genes that have seem to be well associated. And this makes you wonder, okay, can we take this into the animal model, and can we see if it's the same genes, but also get a sense of mechanism? And that's where the research in my lab comes in. We've been looking at HS rats and asking them what would they prefer now. In this case, we're using sucrose solution rewards, so there's some food restriction there. We try to restrict a very small degree. We weigh out animals every day. I think we're at about 90%, but they will do it. They will make choices even if they're at 95% food restricted compared to free feeding weights. Here, as you can see in this example, we have a sucrose reward available in 24 seconds versus some reward now. The reward now is 0 seconds delayed, and we can adjust that based on choices to find equivalence points that are equivalent to our 150 microliters with specific delays. And in our project, what we're looking at is delayed discounting phenotype, typing, collaborating with Abe's group to do genotyping, and then collaborating with Bob Hitzman to do RNA-seq in the prelimbic area, nucleus accumbens core, and BLA, which unfortunately are all limbic regions, as I will show later why that's unfortunate, perhaps. Okay, so these are some data that we've collected. What you can see in this particular slide is the adjusting amount. Remember, we adjust the amount of the immediate alternative until animals start to switch to preferring the delayed alternative. And this slope or gradient marks that switch. So any value of adjusting amount above, they're preferring immediate. Any value of adjusting amount below, they're preferring delayed. Males and females, you don't need any stats to know that there's no real difference there. And I've included some data on the correlations. Here's some examples of some individuals, lots of variability. How to quantify? Well, you can look at the gradient, and that's really useful, but perhaps you need to think more about processes. And so here, with the process-driven approach, you can think about what processes might drive discounting, and they may be opposing. So one model that people have used is looking at a two-component process, present bias, the desire for the reward that's immediate, versus some discounting factor. And as you can see here, for males and females in this sample whose data I just showed you, the females have less present bias. So you'd expect them to be choosing the delayed reward more frequently. However, they have significantly more discounting of the delayed reward, which offsets the less value of the present bias. So you get this push-pull tug of war. And the reason that that's important is that the RNA-seq have suggested all sorts of male-female differences. Here's something in BLA, prelimbic, nucleus accumbens, and the point is not particularly what the genes are, but there's a difference. And if we just use the gradient, we can't see that. But perhaps these differences can be rooted into these other ways of quantifying the behavior. And so I'm pleading in this talk, again, following nicely from Vivek, is that we should really create these sort of behavioral motifs that we link more closely to expression and genotyping and mechanistic processes. And I'd just like to thank everyone for providing animals, etc. And that's all I have.
Lovely. Thanks very much, Suzanne. That's really fascinating. Could I ask a quick question?
Of course.
From an evolutionary angle, what's the selective pressure behind the sort of delayed reward? Is it better something having now because I could be dead later on? What's sort of the received wisdom on that?
So there's been some discussion of that. There's been some hope that, you know, if you're a hunter-gatherer, then maybe as a hunter you want to go for that animal that's wandering around, that deer, whatever. But if you're more agricultural based, then you need to be able to appreciate the delayed rewards, and so that there may be a different selection pressure going on in terms of your job. So.
Yeah, thanks, and thanks for permitting me that very interesting question. All right, great. Any other questions? Please put them in the chat. Thanks very much. We're going to move on to Michelle Parry from the Jackson Laboratories.
Yeah, I should be sharing my screen right now.
Yeah, that's perfect.
Okay, so I'm going to switch gears a little bit and talk more about a resource, in particular looking at how MGI curates strain information we obtain from repositories through the International Mouse Strain Resource. Oops, and it seems to be running away. Okay. So MGI is the authoritative source for mouse strain, allele, and gene nomenclature. We also provide integrated data on gene ontology, gene expression, alleles and phenotypes, and mouse models of human diseases. The goal of our strain curation is to provide standard nomenclature and associate that nomenclature with repository IDs, alleles, and references. We catalog strains we obtain from publication, project loads, direct data submissions, and the International Mouse Resource Center. Users can find information about strains through downloads, the quick search, I'm going to focus on three other areas, mainly the MGI homepage, IMSR, and MouseMine. So there's a lot going on here. This is the strain mini homepage where users can find information about strains by searching for them by name. We also have some other features here that I'm not going to go into. MGI has over 67,000 public strains, which are in over 50,000 of them have repository IDs associated with them. So when a user searches for an ID, they will get a summary page that links to the allele page and as well as back to IMSR, so users can find information about obtaining the mice. IMSR is a catalog of 29 repositories located in North America, Central America, Eurasia, Australia. They have a number of strains cataloged that are live, archived, as well as strains that are ESL only. The way the information gets into IMSR is directly from the repository. IMSR administrator Howie Matenko works with the repositories to standardize the information. Once that's done, we've associated terms like allele, MGI IDs with it. We will then load it into IMSR, and then MGI will retrieve that information from IMSR and check for standardization of the name. So we start out at IMSR with the names provided by the repositories, and then MGI goes through and corrects them. So here's a look at IMSR. You can search by name, allele name, or strain name. This will give you a summary of the alleles. The first column shows if MGI has reviewed the nomenclature. So if it's got a plus, that's approved nomenclature. A minus indicates a disagreement between the repository symbol and MGI's approved symbol. We'll also give the approved symbol. And if there's a question mark, that means that the symbol hasn't been reviewed. And it's up to the repositories to update their strain symbols to match MGI. We actually can't force them to do that. So that's why you'll see some disagreement. IMSR provides links to the repositories and also to MGI for allele information, and that leads back to IMSR as well. The last resource I wanted to talk about was MouseMine. Nice thing about MouseMine is there are a number of queries. You can get queries. You can also design your own queries, all public information at MGI. So I designed a simple query to look for all strains that have repository IDs or external IDs. When you get the result back, you can actually use MouseMine to filter the information to just get the repository that you're interested in. I will end my talk there, and these are just the number of the people who work on MGI's team. Joel Richardson is in charge of MouseMine. Funny enough, I'm the sole strain curator, and how you
to our next speaker, Jesse Ziebarth from UT Health Science Center. Jesse.
Thank you. So today I'm going to talk about the Bayesian Network Webserver, which is something we've developed or called BMW. So you see that on a lot of sites, BMW. This is a website we developed that I think might be something that's useful for some of you to analyze some of your data. Okay, so to talk about BMW, I'm going to go through an example data set and then show you how to use that data set to create a model in BMW. So in my little example here, we're going to say you did some QTL mapping and gave you 5 variables of interest. One of those is a genotype at some QTL. The second is a complex trait or a phenotype that maps to the locus. I'll call that phenotype. So you see phenotype referring to that complex trait. And that could be something like weight if you're studying obesity, or some type of disease outcome after infection. And then then you also have 3 intermediate traits that are associated with that same locus. Those could be things like gene expression traits or immune responses after some infection. And these we'll call trait 1, trait 2, and trait 3. And so once you have this kind of group of interacting variables, but you want to go on to what is the next steps. You want to use these kind of answer questions. and maybe think about things like which trait most strongly impacts that phenotype, which of those intermediate traits most strongly impacts the phenotype. Or if you have other strains of mice, you want to predict what these values of the phenotype and the other traits will be in those strains, or what will happen if you do some experiments and how is that going to impact the other variables in the network. All right, so one way of doing this is use network modeling. And so you're going to create something that looks like this, where you have these arrows that are linking the different variables, where the arrows or the edges indicate interactions between the network variables. And so the first thing you need to do if you're going to use a network modeling approach is you have to learn the structure of the network. You have to decide which of these edges you're going to include in the network model. And you do that based on the data that you use, the data that you've generated. Then you're going to learn the parameters for the network. And then finally, you want to use the network to make predictions. And so we created BMW to kind of integrate all three of these steps so you can quickly and easily create and use a network model once you have your data ready. And so here's an example of this is our little fake data set I've been talking about. Here's the input file you need. So you have the genotype here, where you have A's and B's because there's two genotypes at that locus. And that's a discrete variable. The other variables are continuous variables, where each row here would be a different animal or a different strain. And so next up, I have a video of uploading this input file into BMW and then using it to generate a model. That's something I did in real time, so it's just going to take a couple minutes, and you can use BMW that quickly. But I'm going to skip over some things due to time constraints. So now there might be a couple of things that don't make complete sense because I kind of go over it a little too quickly here. And here's the video here. Here's the BMW homepage. To begin, you're going to click this top left button, load data, begin modeling, and then click choose a file. And then for some reason, the video didn't capture the file manager things, but I'm just loading that file into BMW. And then here you get a little description of the input file that you've uploaded to make sure that that BMW read and interpreted your file correctly. And then we're going to go to this structural constraint interface. And this part I'm skipping over here. And then one thing you can do in BMW is kind of add some constraints to focus on models that are biologically meaningful. So here I'm going to separate this into 3 tiers, where the top tier, kind of the top of the network, is a genotype. The second tier are those 3 intermediate traits, and the third tier is a phenotype. And so now that's going to focus on models where you start at the genotype, go through those intermediate traits, and end at the phenotype. And so this is the model that BMW has learned for that file that we uploaded, for that data set. And so a couple of things here that we can use this model to learn is the intermediate trait that most directly impacted the phenotype here was trait 2. So we get this arrow going directly from trait 2 to the phenotype. And also we see that trait 1 here influences those other 2 intermediate traits based on the data that we loaded. And so at this point, we can go and use this model to make predictions. So we click this button, we go to a different view of the model. It's the same network that we showed previously, just has the distributions of your variable shown. And then if we want to use this to make predictions, what we can do is just go through and click on a variable and then enter the value that we have for that variable. So here, if we have a new mouse strain that we know has genotype B at our QTL locus, we can just click on B here and then hit enter. Now we know what is going to happen to the other variables when we know we have a genotype B for this new strain. And we see how these red lines here are the distributions we predict for genotype B versus the blue line, which is our original distributions considering all of the data. And we can see that once we know we have a genotype B, our model predicts different values for the different variables. And some of them increase, some of them decrease, depending on the relationships between the variables. So a couple of things I want to mention quickly here. You can get quantified values for things like the network parameters and the predictions. Here I just showed the distributions, but you can get actual values here on BMW if you click some of the options on the side. You don't need to have a genotype when learning the model. Here we had 1 genotype. You could have multiple genotypes. This is also used more for relatively small networks with approximately 10 variables or less. It's not something that's going to work for hundreds of variables. The methods we use aren't appropriate for those type of models. We've recently added some new features so you can modify structures or the structure learning settings. And so you can see how these things impact your network structure and how they might impact your predictions. And we also added some features that allow for cross-validation to test the model. And if you can split up your data into test and training data sets, you can use those in BMW. There's also a mirror of BMW that's been integrated into GNetwork. And then here's a couple of examples of places that BMW has been used to look at influenza disease severity in mice and in fungal development. And I think that's all I have for now. Thank you.
Thanks very much, Jesse. Quick question from me, please. How do you decide what's a trait, what's a phenotype?
Yeah, that's something you'd have to you can kind of decide that yourself based on your data set.
Right. What criteria do you use?
Well, yeah, it's going to depend on the data set greatly, but if it's something like gene expression, we know that's kind of something that's simple. It's one direct measure. But if it's obesity, we know that's something that is more complex. There's more factors that go into it than just one single gene, right? But as far as when we're trying to split it up into those tiers and we say, which tier should a different variable go in, that's something that it really is kind of more, you have to kind of use your knowledge of your data set to select better, I guess.
Yeah, I think that Rob just put in the chat about the causality. Sometimes it's more difficult to decide because obesity, of course, can also
right. Yes, exactly. Yeah.
So and sometimes I found myself actually sort of assigning the wrong causality, and I was surprised that that the expression level doesn't link with the transcript level and the protein level, and you're going back to the behavior, so you get the causality the wrong way around, basically. That's an interesting discussion. All right, great. Thanks very much. Any other questions for Jesse? If you could put them in the chat, that'd be great, please. Because again, I've been hogging the question session. Sorry. So can we please move on to our last speaker of the session, Florian Krause from Humboldt University Berlin. Florian.
Yeah. Hello. Thanks for the introduction and welcome to my talk. So my topic is investigating the effect of a large deletion in intron 8 of the BBS7 gene on the Berlin fat mouse phenotype. So the BFMI mouse model is a polygenetic mouse model for juvenile obesity. It is occurred by natural mutation, so it is not a
color-coded for different trait classes. We found 37 traits in pigs with dominant variance above 0.2 and 120 traits in rats and few traits with high dominant variance in mice, which might be because of the low SNP density for HS mice. And notably, we found the hematology and immunology traits tend to have high dominance variance across all these three populations. And then we detect and classified all significant QTLs using ADO. On the left plot here for F2 pigs, each point represents one QTL, and different region from the bottom arm stand for different QTL types, which are additive, partial dominant, complete dominant, and over-dominant. And on average, we identified around 44% of all significant QTLs are showing complete dominance and or over-dominance. And the number here for pigs is around 157, and for rats is around 66, and for mice is around 60 QTLs. And we also found around 40% QTLs were only detected when testing for dominance effectors. And it means these were invisible when we're using the standard additive QTL model. And here, I want to show you an example of the overdominance QTL for T-cell related traits in adjust rats. The first row shows the manipulation using the additive model, and the second row shows the dominant model. And on chromosome 9, there is an overdominant QTL only detected by the dominance model. And the same lobes is also associated with another 3 immunology traits, and none of them could be detected by the additive model. And finally, we also mapped eQTLs across different tissues in each population. Here is one example of F2 pigs liver gene expression data. And when we separate all these eQTLs into different dominant subtypes, we found an interesting phenomenon across all populations, which is the cis-actin eQTLs are predominantly additive. And while the trans-actin eQTLs are enriched for dominant effectors, which suggests that the gene coded for dominant QTLs are less likely to be physically linked to QTLs, but instead act via trans-actin dominant eQTLs. And furthermore, we also find similar conclusions in many different species that people reported before. And at last, I want to express my deeply appreciation to my 3 supervisors. And thank you. Thank you all.
Thank you, Lele, for this presentation. Lovely. Do we have any questions for Lele? Ape's got his hand up.
But if someone else would like, I feel I've asked too many questions.
Yeah, I'm here. Yeah.
Okay. I wonder what is the relationship between the significance threshold you use and the amount of dominance and overdominance you find. And I'm asking because I saw the thresholds you used were a little bit more lenient than I would have used. And of course, if you drop the threshold to something absurd, like 0.05, you would be predominantly have false positives. And then it's very easy to imagine that you would see a different ratio of dominance to additive. And reciprocally, if you made it more stringent, I'm predicting that you would lose a lot of the overdominance and dominance, which might reflect false positives due to an overly lenient threshold. And I wonder if you've looked at just the effect of ratcheting up or down the significance threshold.
Yes, thank you for this is a really good question. Actually, we use the same threshold for all these 4 different QTLs. We just use Bernoulli corrections across them all. And because the SNPs number, especially for the HS mice, is quite low, like we just have 47K SNPs for HS mice. So this threshold is just around 4.5, I think. And
yeah,
also seeing, yeah.
Okay, yeah, I hadn't realized you had so few SNPs. I still think you might want to play with just moving the threshold around and seeing what happens to the ratio of additive to dominance.
Yes, I will try. Thank you. Thank you, Professor.
Great, thank you, Lele. There are more questions for you in the chat if you want to look at those, but we got to move on to our next speaker, Elizabeth Rakowski from the University at Buffalo. Elizabeth.
Okay, everyone can hear me?
Yes, we can.
Perfect. And we can see my screen, hopefully.
Yep.
It feels like mine. Okay.
Perfect.
So I work with Dr. Paul Meyer, which we heard from earlier, like 11 o'clock. And I'm just here to add to basically the project that he talked about, looking at more of the measures that we took during that P50 project. So again, we received our rats from Leah Solberg Woods, and we worked with other universities throughout the country, as well as genotype the DNA. But specifically what I'll talk about is the locomotion and the cocaine condition Q preference that we did, as well as the cocaine contextual conditioning that University of Michigan did.
To expand FP to more.
Sorry. Oh, okay. I don't know if I. Okay. So the Q preference that we measure that we did was They had 4 different trials alternating between cocaine and saline injections, and those injections were paired with different types of tactile floors. So instead of, it's very similar to CPP, so place preference, except for we don't have that distinct spatial location for that. So just for instance, cocaine received the dotted, the hold floors, as well as, and then the saline received the barred floors in this example. We tested them with saline injection and then put them in a box where both floors were available to them. And then we measured how much time they spent on either of those floors. And then we did a post-test for that and compared those. Now, the contextual conditioning is very similar, except for that is basically taking out the saline condition. So we did a pre-test with the saline injection beforehand, 5 trials of the contextual conditioning, say, barred floor with injection of cocaine beforehand, and then we did a post-test with a saline injection. Now, to get into some more Manhattan plots, of course, everybody likes to see those. So this was our main measure, which was the change of time that they spent on the floor that was paired with cocaine. We basically just measured the difference between that by subtracting the pre from the post test time. So the heritability, even though it's slightly lower, we did have a significant GWAS hit on chromosome 19. So there is some locus there that plays a part in this change, which is one of our main measures. And then also 2 other ways that we measured was the distance that was traveled while in those tests. So the distance traveled in the saline condition, which was in the middle of those trials, after a few days of testing, the heritability again is slightly lower, but we did get 2 significant hits on chromosome 6 and chromosome 18. And then on the other hand, distance traveled with the cocaine condition, which is on day 1. So this would be like an acute response to the cocaine. We saw one significant hit on chromosome 6. Now, we also had some PheWAS data. So for chromosome 6, which seems to come up more than once, there also were other phenotypes that other universities and other people in the P50 Center tested for, and that would be rearing in response to the novel stimuli and impulsivity measured by like a reward depletion sequence. So that, I apologize, that was for the CCP. So that was for our Q preference with the 2 different floors. I didn't specify that. So moving on to the contextual conditioning, which is just with the cocaine trials and not with the alternating saline. So the distance traveled in the post-test, so this was the main, I think, result that we see here. There is modest heritability with that. However, you don't see that there really is any significant GWAS hit. So this is basically saying that this is a complex trait. So I guess I'm in the right place. But there is heritability, but there are multiple. This is a polygenic, behavior. So there are multiple genes in here that play some part, but we just weren't able to completely sparse them out. Then we're looking at these other 2 measures. So the distance traveled in baseline. Again, heritability is modest. We did get 2 significant GWAS hits on 2 in chromosome 9 or 18. So this, I would like to say that there is, my theory is that there is like a general exploratory behavior. There's a connection there, like that there is, maybe that's what it was kind of getting at. You see more exploration, maybe you see more impulsivity, like things like that. So that's something that I personally would like to look more into. But again, that's a really abstract theory right now. I don't have evidence of that. So then we also measured head waving, which is a response that you see to like a acute response to cocaine. And there was also a GWAS hit for that on chromosome 16 with lower heritability. Again, we had some PWAS data with this as well. So for chromosome 18, there was association with light reinforcement tests, which is a way that we test sensation seeking, nicotine self-administration, so more drug self-administration behavior, and then some place preference test behaviors as well. Chromosome 16 had some association with elevated the plus maze, so latency to actually enter the closed arms of the maze. So even though there was generally low heritability for these measures, there were some candidate genes that we definitely want to follow up on. So for example, this is the locus plot for the CCP measure. So we have like the large 1 gene, but then right next to it, we have the carboxyesterase and the sodium chloride symporter. And these are also associated with the nucleus accumbens core. So we want to look into the expression or into the QTL for those things. So I definitely think that this laid up a good foundation for us for the future. Did anybody have any questions?
Great, thanks very much, Elizabeth. That's great. Any questions for Elizabeth?
Yeah, I'm going to talk about the GWAS study on the social apparent self-administration in heterogeneous stocks rats. So our result highlights genes implicated in human smoking behaviors. We are part of the P50 study with Dr. Palmer. So some background information, both genetic factors and social factors contribute to cigarette smoking. There are tons of research in human beings. So for us, we established a rat model of nicotine IV self-administration in adolescent rats that acquires social learning between two rats. In this model, self-administrator rats will leak on the active spot to trigger the simultaneous delivery of an oral flavor cue and IV injection of nicotine. Single rats tested using this paradigm will only develop nicotine conditioned flavor aversion. However, present demonstrator rats will consuming the same flavor cue will enable establishment of operant nicotine IV assay. This procedure doesn't require water or food deprivation, so this therefore it's appropriate to study smoking initiation in adolescent. So here we also reported that subject value of the flavor cue doesn't affect nicotine intake. No matter use sweetie oral cue, that is saccharine grape, or bitter cue quinine grape, rats will get the identical infusion of nicotine. So this evidence showed that nicotine intake in this model is driven by the reinforcing effect of nicotine, but not the subject value of the oral cue. So later, we used 12 isogenic strains of rats. We confirmed that nicotine intake obtained in this model is heritable. And we also tested two different doses of nicotine. Most of the strains adjusted their leaking behaviors to maintain the same value, same level of nicotine intake. This dose response further confirmed that the active leaking was primarily motivated by nicotine. So lastly, we identified the carbon disulfide is the mediator of this effect, and demonstrator rats can be replaced by adding carbon disulfide into the flavor cue for the nicotine. So as part of the P50, we conduct GWAS using about 1,600 adolescent HS rats. We first put them through several behavior tests, which Hakan from our lab will discuss later soon. Then the nicotine self-administration procedure will go through 10 days of fixed ratio, followed by 1 session of progressive ratio extinction and cue-induced retreatment of drug-seeking behavior. So we collected the spleen and brains at the end and transferred all the data to Dr. Palmer's group to genotyping and
Fei,
yeah.
Are your slides intended to be advancing? We're still seeing a slide that says social learning supports nicotine IBSA. Okay, now I see GWAS test pipeline. Is that the slide you intend for us to see? Now I see socially acquired nicotine IBSA. I briefly saw one about carbon disulfide, but now I'm not seeing it.
So from on my side, it goes probably to the, right now, it should be GWAS behavior test pipeline.
Correct, that's what I'm seeing now, but I wasn't when I first asked.
Yeah, now I'm processed.
Now it says socially acquired nicotine IBSA and there are 2 figures.
Yes,
Great,
that's true.
Go ahead, sorry.
So now, this is the result of the socially acquired nicotine IBSA. In the HS population, there are a large variation in the nicotine behavior. So this is what we expected. We did a medium split procedure and found about half of these rats showed a preference for the active sport and half of them avoid the active sport. In summary, we analyzed 63 nicotine traits and about half of them have a significant QTL. One of the example is this loci zoom plot. Some of the QTL regions have only one gene.
So I think we're having the same problem. Isn't that right? I'm still seeing socially acquired nicotine IBSA.
So right now should be.
According to the URL, it looks like I'm seeing slide 9, if that's more helpful to you.
So now it should be.
Now I see it, yes.
Maybe my computer is so slow. All right.
Now we have a Manhattan plot.
Okay. Some of these QTL regions have one gene, but most regions have a lot of genes. The QTL regions is 6.9 MBP and have 39 known genes. Here's a summary of our nicotine GWAS. The trait is the number of leaks on the active sport. We on the day 1 trait of nicotine active sport leaking of on the day 1 trait, there is a loci on the chromosome 1 as shown above previous slide. It contains 10 genes that have been reported in human GWAS as well on smoking, alcohol consumption as well. Many of these genes was associated with very similar phenotypes between rats and humans. For example, the WEAT1A, HSPA12A, SHIT1, NRAP, they are all associated with smoking initiation in human being, and our phenotype is the number of leaking during the first nicotine self-administration. So also on the chromosome 16, a region on the chromosome 16 is associated with the numbers of active leaks and nicotine infusion in session 5 and 7. Here is the nicotine infusions.
Thank you. We're still seeing chromosome 1.
Chromosome 1? Okay. So chromosome 1 has like 10 genes that are reported in human GWAS. Here, overlapping with human smoking GWAS. So chromosome 16 is also associated with A5 and A7 actively exposed. In this, this is nicotine infusion. Also, this chromosome 16 loci had associated with nicotine infusion numbers in day 5 and day 7. Moreover, in the total infusion of nicotine, and one gene in this region is TEX29. It is associated with lifelong cigarette consumption in human being, and also GRI4, which is encode a subunit of the glutamate receptor, is a candidate gene for the progression of nicotine intake in our study. And it is also mapped in nicotine dependence in human being. There are several clinical trials that have already used this antagonist for this receptor for alcohol and nicotine dependence. So we want to validate the function of these candidates. Thanks to Dr. G in our department, we can use C. elegans, so that it is cost effective and less time consuming than genetic modifying rats. So he established a model of nicotine conditioned Q preference test in the worm. This is quite similar to the conditioned place preference test in rodents. In this method, worms are placed in a plate containing nicotine and a natural odor called hexane for a few hours. Then they were tested for their preference for hexane in the absence of nicotine.
Thank you. I'm so sorry. I'm not sure whether you wanted to move on. I think we're still seeing slide number 10 or 11 when it says in the URLs number 13.
Right now.
Yeah, so now we're looking at a slide titled Loss of Function of GRI4 impairs nicotine. Is that right? And C. elegans?
Yes, that's correct. So by using this model, we confirm that not all of this GRI4 impaired nicotine conditioned the Q preference in the worms. As you can see in the middle, not all of this gene will abolish the CGP. So this was not, so the GRI4 new mutation doesn't affect their movement or detection of odor by using the IAA test. So many people from our lab participated in the collection and analysis of behavior test. We also sent our collaborator, Dr. Palmer's lab and Dr. Wu's lab provide the HS breeders. Do you have any questions for me?
That's great. I'm very sorry about the problems there. We haven't got much time for questions, but if anyone has any questions, if you could put them in the chat, please, that'd be great. So we can move on to our next speaker, is Hakan Gunturkan, again from the University of Tennessee Health Science Center. Thank you very much again, Tank Fei.
Thank you.
So if you can unshare your slides, then Hakan can upload his.
Yes. So
yes, stop sharing.
Yeah, thank you. Thank you. Can you see now my screen?
Yes, we can.
Yeah, thank you. So hi, everyone. I am also working at Hao Chan's lab at UTHSC. So this is our study. And the personality traits like anxiety, novelty seeking, and social behavior are mentioned to be correlated with addiction in many studies. These all contribute to cigarette smoking in teenagers. And we used the same rats as Tengfei presented. There are totally 1,246 adolescent rats, adolescent heterogeneous stock rats. First, the behaviors that I mentioned were measured, then nicotine self-administration was conducted on the same rats. The open field tests to measure anxiety-like and exploratory behavior, we used an open chamber 1 meter times 1 meter like this. And we used Ito vision to track and record the locomotor movements of the rats. Same type of arena is used for the novel object interaction test with a cylindrical cage in the center. And for social interaction test, there are 2 cylindrical cages. One is empty and one with a stimulus rat that is unfamiliar to test rat. We analyzed the time spent and travel distance in different zones of the fields. In open fields, the frequency of entering the center zone and time spent in the zone is often associated with anxiety-like behavior. Likewise, in novel object interaction test, we are interested in the center zone, which has now a novel object. And in social interaction test, we observe the movements of the rats to the object zone and the social zone to analyze the relation. This is a heat map for phenotypic correlations prepared by using the correlation coefficients. Blue colors here are for positive correlation and red color is for negative correlation. The intensity of the color and the size of the circles are proportional to coefficients of correlation. We put a cross sign like here for 2 phenotypes having no statistical significant correlation. We observe groups of highly correlated phenotypes here. In general, correlations of variables from the same behavioral test are among the strongest. And for example, here, the frequency of visiting the center and the duration of the staying in the center are strongly positively correlated in open field tests. There are also correlations between different behavior tests. Here are some examples of that. For these ones, total travel distances are the highest correlated ones. Actually, we have a previous published study in 2018 for like this, and this is a table from that study. The data sample size was half of the study at that time that we have now. That paper includes behavioral phenotypes and nicotine self-administration. We made a principal component regression analysis, and we found that these behavior phenotypes explains part of the variance in nicotine self-administration behavior. In addition to phenotype analysis, we also did GWAS on these rats. GWAS is done by Abe Palmer's group. Apurva sent us these results actually. The heritability estimates are reasonable, so we expect to find GWAS hits. Indeed, we analyzed many of them, and here is an example of a Manhattan plot and the summary of GWAS hits. And this is a locus plot of significant QTL. To identify the candidate genes for quantitative trait loci, we applied several criteria. One is the presence of a high impact SNP located within the gene. And next is the presence of a significant cis-EQTL in one or more of the 5 brain regions. And third, we check GWAS hits on psychiatric traits. And lastly, we check the expression levels in our RNA-seq data. We identified 13 candidate genes for the traits. Most of these genes have been reported in human GWAS to be associated with psychiatric disorders. So, and lastly, I want to thank to all prior members and current members of our lab and Abe Palmer's lab, Liz Solberg-Wood's lab, and Chenkang Ji lab. So, thank you.
Hakan, that's super. That was actually quicker than the time you got because you ran over the last time. So, have you got any questions for Hakan? I don't see any hands.
Yeah.
Okay, as ever, if you've got any questions, just put them in the chat, please. Great, thanks so much, Hakan, super. And we move
Thank
on.
you very much.
Thank
Yeah,
you.
bye.
We move on to our next speaker, Ellen Reisenberg from UNC Chapel Hill. Ellen.
Can everyone see my PowerPoint?
Yes, and a lovely background picture.
Thanks.
Right.
And can you see the appropriate view here?
Yeah.
Okay. Okay, so my name is Ellen. I'm a second-year PhD student at UNC Chapel Hill in Will Valdar and Marty Ferris's labs. And today I'm going to be talking about some early progress on QTL mapping and collaborative cross mice, which has revealed candidate loci modulating severe coronavirus disease. So as you all probably know, there have been 3 severe epidemics caused by animal to human transmitted coronaviruses in the last 20 years. In 2003, there was SARS caused by the SARS-CoV-1 virus, then Middle Eastern Respiratory Virus or MERS in 2012, and obviously in 2019, COVID-19 caused by the SARS-CoV-2 virus. So an abnormal host immune response, more specifically an overreaction of the immune system, has been indicated in the pathogenesis of severe SARS, MERS, and COVID-19. So given the pathological similarities between the diseases caused by these coronaviruses and the obvious potential for future coronavirus epidemics, it's important to understand the host genetic factors affecting variation and severity of viral disease. So mouse models in general obviously allow us to study infectious diseases in a way that we can't in humans by systematically controlling for environmental variables. The collaborative cross especially provides a powerful system for understanding the genetic architecture of complex traits in general, including viral disease. And the collaborative cross, as you all know, is genetically diverse, capturing 90% of all genetic diversity present in mus musculus. Hope I pronounced that right. Phenotypically diverse across a range of disease phenotypes, genetically well characterized. We have high density genotypes and some whole genome sequencing data for all CC strains. And then as inbred lines, they provide a replicable system, which proved very useful in this context when we were studying original SARS, and then we needed to repeat the study when a much more relevant coronavirus unfortunately came along. So in order to select relevant CC strains for analysis, several strains of interest were infected with SARS-CoV-1, and this was back in 2018 when this was the most relevant coronavirus. And their disease response was measured in terms of weight loss, bleeding in the lung, as measured by a hemorrhage score, and viral titer. So looking at this weight loss plot and removing some of the less extreme responses, we can see that CC6 loses more weight and has more bleeding in the lung, and CC44 and CC23 have less severe disease looking at these same 2 phenotypes. And we can also see that while CC23 has lower viral load, CC6 and CC44 have similar levels of viral load. So equivalent titer but divergent disease response might indicate a dysregulation in the immune response. So we hypothesized that CC6 has an aberrant immune response to the SARS virus and CC44 has a controlled immune response, and that performing an F2 cross between them would shed light on genetically controlled immune-driven mechanisms contributing to this variation that we see in disease severity. So in 2019, we did an F2 cross between CC44 and CC6 and infected the F2 mice with SARS-CoV-1, PBS as a mock infection, and the HKU3 coronavirus, which is another SARS coronavirus in bats that was considered a likely candidate to next emerge in humans. Obviously, SARS-CoV-2 came along first. So in 2020, we repeated the cross and experiment with SARS-CoV-2. And similar to the analysis performed on the CC parent strains, we collected weight loss, viral titer, and lung hemorrhage score information on the infected mice. So ultimately, we infected 1,050 total F2 mice. Almost 400 were infected with original SARS, 275 infected with SARS-2, 90 infected with the non-pandemic HKU3 coronavirus, and almost 300 infected with PBS. I won't talk much today about the PBS infected mice because I haven't really dug into that data yet. Sorry, but I will eventually be incorporating that into my analysis. So a summary of our results so far, I identified several QTL associated with both SARS and COVID-19 pathology. Notably, in the story that I'll focus on today, I identified a peak on chromosome 9 that shows a lot of promise as a putative region affecting coronavirus disease severity, since it has been identified in multiple other studies. It was identified as a QTL in a CC74 by CC11 F2 cross with a similar design. And CC74 and CC11 have distinct founder haplotypes in this region on chromosome 9 from CC44 and CC6. So there appear to be several alleles affecting susceptibility that are segregating there. And then the syntenic region in humans, which is on chromosome 3, has been identified in a severe COVID-19 GWAS that was done in 2020. And the figure from that paper showing this locus is on the right here. And then the haplotype effect seen at this locus in my analysis was observed in SARS-1, SARS-2, and HKU3 infected mice. So the genetic effect at this locus may be applicable to coronaviruses more generally. So taking a look at the F2 data, here we have weight loss time series data for F2 mice infected with SARS-1, including some average trajectories. And then looking at some genome scans, this one is for weight loss on day 1 post-infection, measured as the percentage of starting weight. So as I expected, we don't see any significant peaks. And for all of the genome scans that I'll show, the ticks on the x-axis are informative markers, and the solid and dashed horizontal lines represent the 5% and 10% significance thresholds, respectively, as determined by a permutation test. And then looking at day 2, we see the significant peak on chromosome 9 appear. On day 3, that chromosome 9 peak is more highly significant, and a couple of other significant peaks appear as well. And then on day 4, we see the same 3 peaks as on day 3. And here's a representative genotype by phenotype plot. So this shows the genotype at the marker on this chromosome 9 peak against the percentage of the starting weight on day 4. And this same relationship is recapitulated for all of the peaks shown in these scans, where mice that are homozygous for the CC6 allele have lost more weight than the het mice and the mice homozygous for the CC44 allele. And the average for the het mice being sort of in the middle indicates an additive relationship. And then here's just the same data for the mice infected with SARS-2, the virus that causes COVID-19. The time series weight loss data shows similar trends. Again, on day 1, we don't see significant peaks. On day 2, this chromosome 9 peak again shows up. And on day 3, we still have the peak on chromosome 9, along with 2 additional significant peaks on chromosome 7 and 12. And then those 2 new peaks remain on day 4, and the chromosome 9 peak becomes non-significant, which is interesting, but I don't quite know what's going on there yet. And then again, plotting phenotype versus genotype at this chromosome 12 marker here, we see the same relationship where the mice with the CC6 allele, this marker, have lost more weight on day 4. So here too, with the SARS-2 infected mice, we see the same genotype-phenotype relationship across peaks, with the CC6 allele, as expected, being associated with more weight loss. So the moral of this story is that the results for the SARS-2 experiment recapitulate the peak on chromosome 9 that we saw with the SARS-1 experiment, and we see some additional peaks that appear to be involved with SARS-2 susceptibility specifically. The chromosome 9 locus appears to be playing a role in other aspects of the pathology as well. So looking specifically at lung hemorrhage score, we can see that it's significant at the 10% threshold for the SARS-1 infected mice, and more highly significant with the SARS-2 infected mice. And then the chromosome 7 peak that we saw associated with weight loss and SARS-2 infection is also indicated here. And then just showing the genotype by phenotype plot for the SARS-2 infected mice relative to the genotype of the chromosome 9 peak. This shows that the mice with the CC6 allele have higher levels of bleeding in the lung. And these were the mice that were losing more weight on the previous slide, so we're seeing consistent haplotype effects. And then looking at the HKU3 non-pandemic coronavirus, we don't have the sample size to map QTL, but we see a similar relationship between weight loss and genotype at the chromosome 9 marker that was mapped in the other data sets. So the genotype effect of this peak at chromosome 9 and the mechanisms behind it may be applicable to coronaviruses in general. Conclusions: CC44 and CC6 exhibit divergent disease phenotypes in response to coronavirus infection. QTL mapping in F2 mice from a CC44 by CC6 cross, identifies loci that are associated with this variation. The QTL on chromosome 9 specifically is associated with variation in multiple pathological phenotypes across 3 coronaviruses and has support from other studies in mice and humans. And some next steps for this data, I'll be performing QTL analysis on flow cytometry data that we have from the lungs to see if these loci or any other loci are associated with variations in the concentration of different types of immune cells. And hopefully this would indicate potential immunological mechanisms. And then along with that, incorporating data from the mock infected mice. So if we see a genetic effect on the concentration of a particular immune cell type in both mock and SARS infected mice, that may indicate like a pre-existing immunological phenotype that predisposes one to severe coronavirus disease. And then of course, candidate gene analysis. Sorry. So there are a lot of insights left to be gathered from this data. I'd like to thank my labs, the Valdar lab and the Ferris lab, and then the Barrick lab and the Pardo Manuel de Viena lab for a lot of the mouse work, and my funding sources. And of course, thank you to the organizers, and thank you all for listening to my talk on a Friday afternoon.
Lovely. Thank you very much, Ellen. That's been superb. You were the last speaker today, so really fantastic. Thank you very much. Laura just wants to say just a few more words about the next meeting, or have you done this already in the break, Laura?
Like from the discussion, if you guys weren't able to stick around on the discussion, that there's a lot of mixed feelings about the online versus hybrid versus totally in person. It sounds like, and this is just a summary, that a good mix of those may be the way that most people want to go, or the way to make most people happy. So alternating online and in person, but making some hybrid options available. So I wanted to give the little plug for Boulder, which is still where we're thinking about doing it. If you haven't been to Boulder, it's a whole different place. There's lots of outdoor activities to do. And we could, with 77 out of the 120 people that I think registered, if I'm right, I saw up to about 120 participants at any given time. I think that we would like to do a more formal survey in the coming months and get an idea of not necessarily a commitment to come in person, but a desire to come in person. And that way we have a little bit more direction on looking for venue spaces too. Because obviously if we were doing exclusively online, we'd be looking for 100-person venues. But I have no way to guess on what proportion of that group would prefer to do online. So please, 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 I know surveys are annoying, but when you see one from us come out a little bit later, if you would respond to us, that would help us tremendously in the planning process.
Just a big thank you to Reinmar over the last 3 days. You've done a remarkable job. You're a one-man band. I don't know why we need an organizing committee. That was great, smooth, consistent. And of course to Laura and Abe and Arthur for doing a lot of the legwork for putting this all together. So
thanks, Rob, and thanks everyone. That's been fantastic.
I want to second that, actually, that this has been a terrific meeting and run so smoothly with so little overhead, so little fuss. You know, we put in these abstracts 2 weeks ago. It's the way to do things. It's really, my experience is that it's better than an in-person meeting where there's so much more, you know, overhead of getting there and booking things and submitting your abstracts early.
As one of the organizers here, it's been a great pleasure and a privilege to try and do that. One never quite knows how it works out, especially when you do it on Zoom, and everything was new. So there was no, you know, university conference team behind here. It's basically command control in my office here. It seemed to be working reasonably all right. Thanks everyone for your comments, and we'll take this on board. Please feed back to Laura. It'll be really, really helpful for meetings going forward. So that just leaves me really again to thank everyone for their participation and for your questions, for the interactions. We all had a really great time. It was a very fantastic and useful meeting. We will try and make the recordings available to you, but please bear with me on that, as I will be away on a field course very soon and I can't do anything there, and my media team has said we can't do it because term starts. So it'll be a while. If anyone wants to offer any assistance with that, please drop me a line. It would be great if we could make these recordings available as soon as possible. So we're really trying to make this happen. All right, so it's now about time to close the meeting again. Thank you very much, and hopefully see you in Colorado next year. Thank you very much and goodbye.